So next is spermatogenesis. What is spermatogenesis? Briefly describe the process of spermatogenesis. So what is spermatogenesis? So the formation of a sperm from the primary spermatocyte is known as a spermatogenesis. So the process of formation of a spermatozoa in the male is known as the process of the gamete formation in the male is known as a spermatogenesis. Now this is the process of spermatogenesis through the flow chart. The formation of the spermatogonia from the primordial germ cell. So the multiple number of the spermatogonias are formed. So this process is known as the multiplication process. And after that, the spermatogonias grows in size and form a bigger size primary spermatocyte. So this bigger size cell is known as the primary spermatocyte. And in this primary spermatocytes, the meiosis first division starts, that is a reductional division, and due to that reductional division, the two cells are formed which are known as the secondary spermatocyte, and in these two cells, you can see the number of chromosomes is reduced to the half. So here this is particular diploid cell, and these two are the haploid cell structures. So which is having a single set of chromosomes. Now in this secondary spermatocyte, the meiosis second division continues, and this will be a equational division. So there is no reduction in the chromosome number, and ultimately the spermatids are formed. These spermatids will then transform into the sperm through the process of spermiogenesis, and this completes the formation of spermatogenesis. So this particular diagram is explaining the complete process of spermatogenesis. Now coming to the next. Name the hormone involved in the regulation of spermatogenesis. So what are those hormones which are helpful in the regulation of the spermatogenesis? So we already know that at puberty, the hypothalamic hormone, that is GnRH, gonadotrophin releasing hormone, start releasing, and due to this, it will affect the anterior pituitary. It will stimulate the anterior pituitary for the secretion of gonadotrophins. Then these gonadotrophins are known as a LH, that is luteinizing hormone, and FSH, that is a follicle stimulating hormone. Now that LH will reach the Leydig cells inside the testis and stimulate those Leydig cells to form the androgens, or then we can also call them as a testosterone. So androgen or testosterone, one and the same thing. Now, so these four hormones are helpful in the regulation of the spermatogenesis. These androgen released by the Leydig cell will stimulate the spermatogonia, and spermatogonia after is stimulating forms the sperms. So define the spermiogenesis and spermiation. There are two processes, spermiogenesis and spermiation. Spermiogenesis, I have already told you, like this in this particular diagram we can see that spermiogenesis is a process from the conversion of spermatid to the sperm. This particular conversion or transformation of spermatid from the sperm is known as a spermiogenesis. So this is the particular spermiogenesis process. The process of transformation of the spermatid into spermatozoa is known as in the seminiferous tubule is known as a spermiogenesis. Now once the spermatozoa are produced, these spermatozoa are still getting nutrition from the Sertoli cells. So that is why for the purpose of nutrition, the head of a spermatozoa is still embedded inside the for the nutrition in the Sertoli cells. So after spermiogenesis, the sperm head become embedded in the Sertoli cells and finally released from the seminiferous tubule by the process of the spermiation. So until when the particular amount of the nutrition has been obtained, the spermatozoas get released by the seminiferous tubules or the Sertoli cells into the lumen. So this particular release of spermatozoa from the seminiferous tubule is known as a spermiation. So there are two different processes, spermiogenesis and spermiation. Coming to the next, what is the label diagram of the sperm? Draw a label diagram of a sperm. So this is the NCERT diagram. We have already talked about this. That particular sperm has been classified, has been divided into the four parts. That is the head, this part is the neck, this is middle piece, and then the tail part. So the particular sperm is classified into the four different parts, that is head, middle piece, neck, and the tail part. So head is, again, if we talk about having the nucleus containing the chromosomal material, the anterior part of nucleus is having a acrosomal cap, acrosomal structure which is containing the sperm lysins helpful in the fertilization process. The whole sperm is a haploid cell which is covered by a membrane that is known as a plasma membrane. This constriction between the head and middle piece is known as the neck region, which is containing the centrioles, and in the middle piece, the particular neck, the middle piece, the particular spiral arrangement of the mitochondria, that is known as the Nebenkein sheath, which is the energy source and helpful in the movement of motility of the sperms. Now then the tail portion, it is the longest part. It is made up of the axonema or axial filament, which is raised by the centriole. So this is the particular structure of a sperm we have already talked about. Now the next question, what are the major component of the seminal plasma? Before that, what is seminal plasma? So when we come to the seminal plasma, the major component of the seminal plasma are, so secretions of the accessory sex gland of the male, means all the accessory sex gland of a male when collectively secrete a fluid, that particular fluid is known as a seminal plasma. It means the secretion of the prostate gland, secretions of the seminal vesicles, secretions of the bulbourethral gland, all the glands, the collective fluid released by all these glands is known as a seminal plasma. And what does that seminal plasma is mainly having? So mainly it is composed of the calcium, fructose and certain other enzymes. So this is the particular composition of the seminal plasma. Now, what are the major functions of the male accessory ducts and glands? Means all the major accessory ducts of the male and major accessory glands of the male. What are these functions? So, the major function of the male accessory ducts and glands are as follows. Now, the male accessory ducts includes retrotestis, vasa efferentia. So, vasa efferentia, it conducts the sperm from retrotestis to the epidermis, means outside the testis in the long coiled tubular structure that is known as the epidermis. Retrotestis, it stores sperms that are produced by the seminiferous tubule. So what is retrotestis? It is a dense network of a tubule. When the seminiferous tubules from all the testicular lobules carry the sperms to the posterior surface of the testis, they form a dense tubular network, and that dense tubular network will be known as a retrotestis. So it is a particular structure in which the sperms are collected. So store sperms that are produced by the seminiferous tubule, and from this retrotestis, the sperms are carried by the fine ductules, which are known as the vasa efferentia, and these fine ductules will carry sperms to the epidermis. Now, vast difference. Conducts sperm from epidermis to the urethra. So it conducts the sperm from the tail part of epidermis, the vas deferens arises, and it carries the sperms to the abdominal cavity. Epidermis, the physiological maturation of the sperm, the longest part of the particular accessory duct, the longest accessory duct is the epidermis, and this epidermis is made up of three parts: head, body and tail part. So the maturation of sperms takes place inside the epidermis, storage of sperms takes place mainly inside the, also we can say the storage of sperms takes place inside the epidermis, and certain particular secretions from the epidermis helps in the nourishment of the sperms also. So now, male accessory glands. So there are three different male accessory glands: seminal vesicle, prostate and the Cowper's gland. The secretions of the seminal vesicles activates and provides energy to facilitate sperm motility after the ejaculation. So after the ejaculation, the energy that is needed by sperms to show its motility is provided by the seminal vesicle, mainly fructose. Now, prostate gland nourishes and activates the sperm. It also nourishes, the secretion of the prostate gland also nourishes the sperm and activates the sperms, enhancing the sperm motility. So after the ejaculation, they are also helpful in the enhancing the sperm motility of the sperm. Cowper's gland, it enhances the mobility and survival potentiality of sperms in the genital tract of female reproductive system. The genital tract of the female reproductive system, in that particular genital tract, it helps the sperms to survive. So Cowper's gland secretions are the alkaline secretions which help the sperm to survive the female reproductive tract. It enhances the mobility of the sperms also, and neutralizes the acidity of female vaginal secretion. So it also neutralizes the acidity because female particular vaginal secretion. So particular genital tract of a female is acidic due to the secretion of the due to the normal secretions. हटाओ यार इसको यार, ठीक है साला क्या लिख दिया है यार, कहाँ से लेके आया है. So now the male accessory glands. Male accessory glands include seminal vesicle, prostate gland and the bulbourethral gland. The seminal vesicles activates and provides energy to facilitate sperm motility after the ejaculation. So when the sperms are ejaculated outside, the motility of sperm is obtained because of the secretions of the seminal vesicle. Similarly, the prostate gland, it nourishes the sperms and it also activates the sperm. It also helps in the sperm motility. Cowper's gland secretion, it enhances the mobility. So all the secretions enhancing the mobility of a sperm, and it also enhance the survival potentiality of a sperm in the genital tract of the female. So when the sperm are ejaculated in the female genital tract, the potential of a sperm, the survival potential of a sperm is enhanced by the Cowper's gland alkaline secretions. Now, it also neutralizes the acidity of the vaginal secretions, means the female vagina is secreting the acidic content, and that acidic content is not favorable for the sperm. So, that particular acidity of the vaginal secretion is neutralizes by the secretion of this Cowper's gland and the bulbourethral, we can also say. Now, the third question is, what is oogenesis? Give a brief account of oogenesis. So, when you talk about oogenesis is again a phenomena of the formation of the female gamete known as ova from the diploid oogonia in the ovary, called as a oogenesis. So, similarly like the spermatogenesis in the male, in the female, the process of the gamete formation is known as a oogenesis. Now, oogenesis can explain through this particular flowchart. So, this I have already showed you and already explained you this particular flowchart, that the oogenesis process, the particular first division of the primordial germ cells up to the fetal life, the 7 million oogonias are produced through the mitosis. So, the number of oogonias increases to the multiple folds. So, this particular division is known as a multiplication phase, when the primordial germ cells converts into the millions of the oogonias. Now, these oogonias will get nutrition and they will grow in size. So, when they increase in size, they will form a slightly bigger cell that is known as a primary oocyte. In this primary oocyte, the meiosis begins and arrests in the prophase of the meiosis, prophase first of the meiosis first. Now, after the puberty, this particular primary oocyte, the meiosis first division get completes, and this particular cell is divided into the two different cells. One is a bigger cell, that is a secondary oocyte, and one is a smaller cell, that is the first polar body, that usually degenerates, or sometimes it may also get divided. So, it usually degenerates. Now, the secondary oocyte enters the meiosis 2 and this enters the meiosis second, and the meiosis second is also arrested in the metaphase second, and then in this arrested phase, this secondary oocyte is released by the ovary, and when it is released by the ovary, it will reach the ampulla of the fallopian tube, where it waits for the arrival of the sperm. If the sperm arrives and penetrates this will continue to divide, and this will resume its meiosis second division, which was arrested in the metaphase 2, and again form the two cells. One is the smaller cells and the bigger cell. So, the unequal cytokinesis taking place. One is a smaller cell, that is known as the second polar body. The one which is bigger cell is known as a ovum, or it is also known as a oocyte. So, the second meiosis division only resume after the penetration of the sperm. If the particular sperm does not penetrate the secondary oocyte which was placed inside the ampulla of the fallopian tube, the secondary oocyte get degenerate in this particular stage. So, as such, the secondary oocyte will not continue the meiosis second, and it will degenerate as such. So, this is all about the oogenesis process. Now, draw a label diagram of the section through the ovary. So, this is the cut section of the ovary. So, diagrammatic section of the ovary is, this is the particular thin epithelium which is covering the matrix of the ovary. The complete matrix inside this particular structure is known as the matrix, or is also known as the stroma of the ovary. This stroma is again divided into the middle part, that is medulla, and the peripheral part, that is known as the cortex. And we can see that in the cortex part, the follicular genesis is taken place. So, the follicular genesis has taken place inside the cortical portion of the ovary. So, this is the cut section or the cross section of the ovary. Now, draw a label diagram of the Graafian follicle. So, a mature follicle or a Graafian follicle. This is the diagram I have already explained. This in particular diagram, we can see that the size of a Graafian follicle is increased by multiple folds due to the presence of this cavity which is known as the antrum, and due to the presence of this antrum, all the granulosa cells will become thin, and they will just go to the periphery. They will just arrange at the periphery, and due to this oocyte which was attached to these granulosa cells also become eccentric. It goes to the corner of the particular structure, and during the mature Graafian follicle, the meiosis first division has also completed. So, this oocyte will become the secondary oocyte. So, the secondary oocyte is seen in the mature Graafian follicle stage. In this particular oocyte, this outermost this covering of red color is known as the zona pellucida, which is covering the secondary oocyte. And outside this zona pellucida, we can see there are some corona radiata, there are some granulosa cells. These granulosa cells which are present outside the secondary oocyte or zona pellucida are known as a cumulus oophoricus. These are known as a cumulus oophoricus. And out of these cumulus oophoricus, some of the cells of the cumulus oophoricus which are present in the close vicinity close vicinity of the zona pellucida or the secondary oocyte get elongated, and these cells of the cumulus oophoricus which are present in the approximation or the close proximity of the zona pellucida are named as a corona radiata. They are known as a corona radiata. There are some more cells which attach these cumulus oophoricus to this peripheral granulosa cells. So these cells in between these cells are known as a discus proligerus or the germ hair. So this is the complete structure. In this structure we can see that there is a clearly arranged theca interna and theca externa layer. The outermost layer, the fibrous layer, is known as the theca externa. The inner cellular layer is known as the theca interna. Now after this we can see the name, the function of the following: corpus luteum, endometrium, acrosome, sperm tail, and the fimbriae. So first of all, corpus luteum. What is corpus luteum? So the corpus luteum is a yellow colored structure and secretes the progesterone hormone, mainly progesterone hormone, during the luteal phase of the menstrual cycle. So this progesterone is helpful in the maintenance of the endometrium. So this is corpus luteum. Now coming to the endometrium. The endometrium is the innermost layer of the uterus comprising of the glands that undergoes the cyclic changes during the different stage of the menstrual cycle in order to prepare itself for the embryo implantation process or the possible pregnancy. So endometrium is a particular innermost layer of the uterus which undergoes the cyclic changes during the menstrual cycle. Now coming to the next, acrosome. So acrosome is a cap-like structure present in the head portion of the sperm just above the nucleus, and this acrosome consists of the sperm lysine enzymes. These sperm lysine enzymes are helpful in the penetration of the outer membrane of the egg. So these enzymes are helpful in the penetration of the outer membrane of an egg by the sperm, which facilitate the sperms to perforate the outer membrane, and it enters the particular outer membrane, and then it will reach the plasma membrane of an egg, and then it will fertilize with the egg. So they help in the fertilization process. Now sperm tail. The sperm tail makes up for the longest part of the sperm, enabling the movement of the sperm once it has entered the female reproductive system. So once after the ejaculation, the sperm needs its tail for the movement to reach the female ovum. So when it has, in order to reach the female ovum to fertilize it, the motility of the sperm is gained by the sperm tail. Now fimbriae. So towards the ovarian end, so in the particular female reproductive structure, we have seen that the fallopian tube is having three parts: isthmus, ampulla, and the infundibulum, and the terminal part of the infundibulum, which is towards the ovary. So the particular part of the fallopian tube towards the ovary end of the fallopian tube, finger-like projections are arise. These finger-like projections are known as a fimbriae. These are particularly known as a fimbriae, which are assist in the collection of the ovum after the ovulation. So when the ovary is releasing its secondary oocyte, this secondary oocyte is get collected by the finger-like projections known as fimbriae, and then the secondary oocyte is transferred to the ampullary region of the fallopian tube. So these are all the functions. Now the true-false statement type question. To identify the true-false statement, correct each false statement to make it true. So we also should know the correct statement also. So first is androgen are produced by the Sertoli cells. So androgen is produced inside the male body. So they are inside the, we talk about the testis. Inside the testis, the androgen are produced, the male sex hormones, but they are produced by Sertoli cells or Leydig cells. So this statement, the androgens are produced by Sertoli cells. This is true or false? So this will be the false statement because the androgens are mainly produced by the Leydig cells. Now the second question, that is the spermatozoa get nutrition from the Sertoli cells. So we all know that Sertoli cells are the nurse cells which provide nutrition to the developing stages of the sperm. So spermatozoa also get nutrition from the Sertoli cells. So this is the true statement. So we need not to get any more statement with any correct statement because the given statement is correct. Now Leydig cells are found in the ovary. So Leydig cells are found in the ovary. Leydig cell is a part of the male reproductive system, and ovary is a primary reproductive organ of a female. So can Leydig cell be a part of an ovary or not? So this is the false statement because Leydig cell cannot be a part of an ovary. It is a part of a male reproductive system. So the correct statement would be Leydig cells are found in the testis of a male reproductive system. Now next, Leydig cell synthesizes androgen. So this is the true statement. Leydig cells are synthesizing the androgens which will stimulate the mitogonia for the spermatogenesis. Oogenesis takes place in the corpus luteum. So oogenesis takes place inside the corpus luteum or it takes place inside the entire ovary. So oogenesis is a process which takes place, it is a false statement. Oogenesis is taking place inside the ovary, and this will lead to, the ovulation will lead to the formation of the corpus luteum. So both structure are present inside the ovary. Particularly the oogenesis process is taking place inside the ovary, and corpus luteum also present inside the ovary. So the process of oogenesis, the gamete formation, the process of the female is taking place inside the ovary. It is not taking place inside the corpus luteum. Now, what is the menstrual cycle? Which hormone regulates the menstrual cycle? So we all know that menstrual cycle is a reproductive cycle of the female primates like monkeys, apes and humans. In female, the menstruation is repeated at an average of 28 to 29 days, and the time between the one menstruation to the next menstruation is known as a menstrual cycle. Now, which hormone regulates the menstrual cycle? So we have already told you in the detail that the hormone that takes place in the regulation of the menstrual cycle are the gonadotrophin hormone, the FSH, LH, progesterone and the estrogen. So hypothalamic hormones also takes place in the regulation, which is not written here, but hypothalamic hormone which stimulate the secretion of LH and FSH from the anterior pituitary. So LH and FSH are given, and then this LH and FSH, the pituitary hormone, when this will stimulate the ovary for the secretion of the estrogen and the progesterone. So these all hormones are regulating the menstrual cycle. So they are used in the regulation of the menstrual cycle. Now, what is parturition? Which hormones are involved in the induction of parturition? So what is parturition? So when the baby gets fully developed, so parturition is a process wherein the fully developed fetus from the mother's womb is expelled out after the completion of the gestation period, means after the completion of the embryonic development, the completion of the 9 months inside the female womb or the uterus, we can say. So after that, when the fetus get fully developed, this fetus is expelled outside from the mother's body, and this process of expelling this fetus outside the female body is known as a parturition. Now, there are two critical hormones that are involved in the induction of the parturition. So, these two critical hormones are, the first is the main parturition hormone, that is the oxytocin, and second one is also a supportive hormone, that is the relaxin. So, oxytocin, what does it do? So, it directs the full-term fetus towards the birth canal. Birth canal is the vaginal canal, or we can also say the cervix plus vaginal canal is the birth canal. So, it directs the full-term fetus towards the birth canal of the female body. So as it causes the contraction of the smooth muscles of the myometrium of the uterus, the baby was pushed toward the cervix, and it has ultimately been expelled outside by the female or the mother. Now the second hormone, that is relaxin. Relaxin, as the name suggests, it relaxes the pelvic ligament or the pelvic girdle, and widening helps in the widening of the pelvis area of the female. So it is making a particular space available for the fetus to get expelled out. So it is widening the pelvis to assist in an easier childbirth. So when the more space is available to get the expellation of the baby will be delivered easily. Now, in our society, the women are often blamed for the giving birth to the daughters. In our society, the women are often blamed for the giving birth to the daughters. Can you explain why this is not correct? So, is it correct to blame a woman for having a male child, for delivering a female child and not the male child? So now, this is not correct to blame a woman for giving birth to the daughters. So it is not correct. Why it is not correct? Because we have already told you that the sex determination, during the sex determination, the father is responsible for the determination of the sex, not the mother. So the male sperm contains either X or Y chromosomes, whereas the female egg only contains the X chromosome. And for the son, the Y chromosomes is necessary. It is responsible. For the formation of the male boy, the Y chromosome is responsible. So the female is not having any Y chromosome. So how can we blame female for that? So only the male sperm is containing either X or the Y chromosome. So only male should be blamed for this not having a son child or a son or the male child. At the time of fertilization, the sperm with the Y chromosome combined with the egg containing X chromosome formed, which will be forming the male. Now, thus scientifically, sex of a baby is determined by the father and not by the mother as blamed in our society. So the father should be blamed, the mother should not be blamed for not having a son child. Now, not having a male child or a son. How many eggs are released by a human ovary in a month? How many eggs do you think would have been released if the mother gave birth to identical twins, would your answer change if the twins born were fraternal? So first we have to understand what is identical twins and what is fraternal twins. There are two terminologies. They are identical twins and they are fraternal twins. What are these? So I will be going to explain with the diagram. So normally we can see that typically in a month the ovary will form a one gamete. So human ovaries release only one gamete. But sometimes due to certain reasons, certain unknown reasons, it also can release two gametes at a time. In those cases, when the two gametes are released, if more than one sperm penetrate, if the two sperms penetrate each of the gamete, means the two gametes are, normally the one gamete was released which was fertilized by the one sperm. If two gametes are released and both the gametes, both the ovas are fertilized by the sperms, so different sperms, so these two different ova and the fusion of the two different ova with the two different sperms will form the two different zygote. So the two different zygote will be going to develop inside the female body, and then the female is giving birth to the twins. So now this is the two, there are two different conditions in case of the identical twins or the monozygotic twins. So one is the identical twins, which is also known as the monozygotic twins, in which the one egg is released by the ovary which splits into the two post fertilization. This is why the identical twins exhibit the same genetic feature. We will come into this. On the other hand, the fraternal twins or the dizygotic twins, two eggs are released which are fertilized by the two different sperm, causing the fraternal twins to exhibit different genetic characteristics. Now coming on to this, through this diagram we can see there are two different type of twins, the monozygotic twins and the dizygotic twins. Normally a female produce only one ovum. This is the one ovum that is produced by the female. This is one ovum that is produced by the female. If the female is producing one ovum, any one sperm can fuse with this ovum and form a zygote. So only one zygote is formed. So when the one zygote is formed, this is a monozygote. Only one zygote, so this can be called as a monozygote. And after development, during the cleavage division, this particular zygote splits into two. So when the zygote splits into two, these two cells, or I can say these two blastomere, will give rise to the two different organisms. It means when a single zygote splits due to uncertain region, we don't know the region, when the single zygote splits during the cleavage division, the two blastomere will be giving rise to the two different organisms. So in this condition, these two different cells are genetically identical because they are developed from the single zygote. They are developed by the fusion of the one male sperm and a one ovum. So they are genetically identical. These two cells are genetically identical. Therefore, the organisms that are developing from these two cells are also known as a genetically identical organism. So they are also known as the identical twins. So they can share their placenta or they can separate, the placenta can be separated. So this is not usually seen. So sometimes there can be a separate placenta for both the babies, or they can be shared placenta for both the babies. So this is all about this. Now coming to the dizygotic twins, if a female, normally female releases one ovum, but if a female releases two ovum, then these two ovum, this is ovum 1 and this is ovum 2, these two ovum will fuse by the two different sperm. You can see the difference between this. This is a light pink in color, this is light yellow in color. So this is a different sperm fusing with the ovum number 1, and this is the different sperm which is fusing with the ovum number 2. So when the two different ovum fuse with the two different ovum, they will form two different zygote. This is also a zygote and this is also a zygote. This is zygote 1, this is zygote 2. So when the two ovums were released, the two zygotes were formed. These two zygotes will give rise to the two different blastomeres and two different organisms. But these two different organisms, in these two different organisms, the ovum and sperms are different. So that is why they are genetically not identical. They are genetically distinct, but they share the particular gestation period, because they are developed together. So they will take the same 9 months inside the mother womb. So they will develop together through that 9 months period, but they will be having a separate placenta always. So these two babies, which will develop from the two different ova and the fusion of two different ova and sperm, and forming the two different zygotes. So that is why these two organisms are known as twins, but they are only dizygotic twins. They are developed from the two different zygotes. This is developing from, assume that this is developing from this zygote, and this will be developing from this zygote. So when they are developing from the two different zygotes, they are known as dizygotic twins. So this is the difference, the monozygotic twins and the dizygotic twins. In the monozygotic twins, female is only releasing one ovum which is fused with the one sperm, but afterwards, when the zygote was formed, when the one zygote was formed, this zygote splits into the two different cells, and each blastomere will be giving rise to a different organism. So the splitting occurs after the formation of a zygote. So that is why there the identical twins were formed, and here the two different ovums are released by the female, which is fused by the two different sperms. So the two different sperms will be genetically not similar, and two different ovums were also not genetically similar. So the organism, the zygote produced from them is different. So the organism producing from these two different zygote will also be distinct. So they are genetically distinct and giving rise to the two different genetically distinct twins. So this is known as a dizygotic twins. These twins are developing from the single zygote. These twins are developing from the two different zygotes. So they are known as the monozygotic twins. They are known as a dizygotic twins. So in condition of the monozygotic twins, they are genetically identical. In condition of the dizygotic twins, they develop from the two different zygotes. So they are not genetically identical. So this is all about. Now, how many eggs do you think were released by the ovary of a female dog which gave birth to the 6 puppies? So normally in the human we can see only the 1 or 2 babies are developed, but in the other organisms, other related organisms like dogs and cats and other organisms, we can see that the more than 1 puppies or more than 1 organisms are developed, the offsprings are produced. So if in a dog, if 6 puppies are produced, how many eggs do you think are released by the female, that particular mother of those dogs? So we can see, in order to have given birth to the 6 puppies, the ovary of the female dog released 6 different eggs. Hence 6 zygotes were formed for each to develop into a puppy. So for the formation of the 6 puppies, the 6 ovums or 6 different gametes are released by the ovary of the female dog. Now this is all about. Now some previous year questions are also there. So in CBSE 2020, there is a 3 marks question. A case-based question has been asked. Study the given diagram where A is an embryonic stage that get transformed into the B, which in turn get implanted in the endometrium in the human female, and we have to identify that A, B, C and D. So what are A, B, C, D? Write the name of the cavity present in the stage B. So this is the B stage and there is a cavity present inside that. We have to name that cavity. State the fate of C and D in the course of embryonic development in the human. So we have to also explain the fate of the C, means these particular cells which are hanging inside, and these outer peripheral cells. How will they transform into that particular structures? We have to tell them also. So now the answer for this question is A is so this particular structure we can see there is a 8 to 16 cell structure. So A will be the morula. So this morula will divide and transform into a particular structure, which is, I have already told you, the multicellular structure. So B will be the blastocyst. So this blastocyst, A and B. Now what is C and D? A blastocyst consists of 2 type of cell. First is the outer peripheral cells, means D, and the inner cell mass, that is the C. So now the innermost cells which are hanging, which are attached with this particular outer cells, is known as the inner cell mass. So C is the inner cell mass, and outermost peripheral cell, that is a D, is the trophoblastic cells. So the morula is A, B is blastocyst, C is inner cell mass, and D is a trophoblast. Now they are asking about the cavity present between, name the cavity present between the stage B. So this is the blastocyst stage, and the cavity of the blastocyst stage is known as a blastocele. So the cavity present inside the stage B is a blastocele. Now state the fate of C and D in the course of embryonic development in the human. So what will the C and D transform into? So C is the inner cell mass, means they will be transforming into a complete embryo. So inner cell mass differentiated as an embryo in the further development. Now and D is a trophoblastic cell, and trophoblastic cells helps in the implantation and it also helps in the development of the placenta. So the transformation of the trophoblast into the placenta in the further development. Now this question is over. Now the construct a flow chart exhibiting the sequential events of the oogenesis. So this particularly asked in the CBSE 19 to 2019, and 3 marks question, short answer type 2 questions. And in this we have again the same diagram that I have already told you. So you have to make this diagram and to explain this diagram, that is. Now given below the diagram of a human ovum surrounded by the few sperms. Study the diagram and answer the following question. This is asked in the CBSE 19 2019, 5 marks long answer type question. So in the 2019 different zones are there, so some more questions can be from a 1 year in a particular different paper. So again 2019 questions, 5 marks question, long answer type question. In this particular diagram we can see that some of the sperms are surrounded, the particular ovum is surrounded by some of the sperms. Now they need to ask some questions like which one of the sperm would reach the ovum earlier. So we can see this diagram and we can find out that which of the sperm is going to penetrate the ovum out of these A, B, C, D and E. So the one who is closer to the ovum will reach the ovum, which will penetrate the ovum first of all. So the A will be the answer for this question. Which of the following sperm will reach the ovum earlier? That is the sperm A, because it is closer already. Identify D and E. What is D and E? So this D is the outermost layer made up of the granulosa cells, or I can say the corona radiata cells hanging around the ovum. Now what is E? So this particular E, this line is coming here as up to here. So this particular E is a zona pellucida. The green color cell which we can see outside the ovum is known as the layer of the glycoprotein outside the ovum is known as a zona pellucida. So the D is the cells of corona radiata and E is a zona pellucida. Mention what helps the entry of sperm into the ovum and write the changes occurring in the ovum during the process. So what helps the sperm to penetrate these two layers? So the secretion of the acrosomes or acrosomal enzymes, which is also known as the sperm lysins present inside the acrosome, which help in the entry of a sperm into the ovum, induces the completion of the meiotic division of the secondary oocyte. Means before the penetration of ovum there was a secondary oocyte, and when the sperm comes in the contact with that secondary oocyte, it transforms the secondary oocyte into the ovum by completing the meiosis second division. So these are the two changes. The secretion of acrosome helps in the entry of the sperm, as well as the entry of the sperm induces the meiotic second division for the completion of the conversion of the secondary oocyte to the ovum. Now ampulla and ampullary ischemic junction, for what this is asked for. The name the specific region in the female reproductive system where the event represented in the diagram takes place. So what is this event? The sperms coming in contact with the male gamete is coming in contact with the female ovum. The male gamete and female gamete fusion. So this fusion of the male female gamete is known as a fertilization. So this event of fertilization. So they are asking name the specific region in the female reproductive system where the event represented in the diagram takes place. Means they are asking about where the fertilization taking place. So fertilization takes place inside the ampulla or ampullary ischemic junction of the fallopian tube. So this is the last answer. The ampulla and ampullary ischemic junction. Both are correct. Now medically it is advised to all young mothers that breastfeeding is the best for the newborn babies. Do you agree? Give reason in support of your answer. So the breastfeeding is good or not for the baby. So this is asked in the CBSE 2018 short answer type 2 question, 3 marks question. So do you agree with that the young mother should breastfeed their newborn baby or not? So yes, the mother should breastfeed their baby because the milk produced during the initial few days of the lactation is known as a colostrum. So after the birth of a baby, the milk produced by the initial few days of the female is known as a colostrum, and this colostrum contains several antibodies absolutely essential to develop the resistance inside the newborn babies. Breastfeeding during the initial phase, initial period of the infant growth is prescribed or recommended by the doctors bringing up the healthy baby. So this is all about. State from where do the signals of the parturition originate in the human female. So from where the signals of parturition arises. So the first signal of the parturition arises from two parts, that is a fully developed fetus as well as the placenta. So the fully developed fetus and placenta are from there the first signals of the parturition are originated in the human female. Now, explain the menstrual phase in the human female. State the level of ovarian and pituitary hormone during this phase. Explain the menstrual phase in a human female. State the level of ovarian and pituitary hormone during these phases. So first of all, we have to talk about the menstrual phase, and menstrual phase is the first phase, that is we can also say the bleeding phase. So they are asking that explain the bleeding phase and also the amount of hormone, the level of hormone during that phase. So second is why the follicular phase in the menstrual cycle is also referred to the proliferative phase. So why the follicular phase is also known as the proliferative phase, and explain this. Explain the event that occur in the Graafian follicle at the time of ovulation and thereafter. So what are the events taking place inside the Graafian follicle at the time of ovulation and after that? And draw a Graafian follicle, label antrum and a secondary oocyte. So this is a 5-mark question asked in the CBSE 2016, and it is a long answer type question. Now, first of all, menstrual phase occurs when the released ovum not fertilized. So when the pregnancy do not take place. Breakdown of the endometrium. So when the pregnancy was not taking place, the corpus luteum was not stable. Corpus luteum will start degenerating. Due to the degeneration of the corpus luteum, there is a low level of progesterone and estrogen. When there is no progesterone or the less amount of progesterone, the endometrium cannot be maintained, and if the endometrium cannot be maintained, the breakdown of the endometrial lining of the uterus takes place. So when this breakdown takes place, with these blood vessels, this forms a liquid that comes out through the vagina, which is known as a menstrual flow, and it lasts for the 3 to 5 days. So this is all about the bleeding phase of the menstrual phase. Now, what is the level of hormone? So during that phase, when there is no corpus luteum, there is no ovarian hormone, and as well as this particular phase is having a no significant rise in any of the hormone. So all the hormones, whether it is the ovarian hormone or whether it is pituitary hormone, both the hormones, the level of all the hormones from the ovary as well as the pituitary is low. So the low level of ovarian and pituitary hormone during this particular phase. Now in the ovary, the follicular development. So why the follicular phase is also known as the proliferative phase. So we can say the follicle during the particular phase when the follicular development was taking place inside the ovary. At the same time, inside the uterus, the development of endometrium taking place through the proliferation. So this simultaneous effect on the ovary and the uterus will be responsible for the name given to the follicular phase and the proliferative phase. If we're talking about the ovary, the follicular development is taking place, so it is also known as the follicular phase. If we talk about the uterus at the same time, the proliferation of the endometrium is taking place, so it is also known as a proliferative phase. This is the answer. Graafian follicle ruptures to release the ovum. So what happens in the Graafian follicle? Explain the event that occurs in the Graafian follicle at the time of ovulation and after that. So what happens in the Graafian follicle? So at the time of ovulation, the Graafian follicle ruptures and releases the ovum, or it is also known as the secondary oocyte at that time. So it releases the secondary oocyte, and what occurs after that? So remaining part of the Graafian follicle will transform into the corpus luteum. So a new structure was formed inside the ovary, that is the corpus luteum, and the Graafian follicle get ruptured and it releases the gamete which will reach the fallopian tube for the expected fertilization. Now, and this is asking about the last question about this. Draw a Graafian follicle, label antrum and the secondary oocyte. So this is we have already got discussed with this. This is the particular structure of the mature Graafian follicle, and in this structure, this is the secondary oocyte, and we can also figure out this particular cavity from here to here. This is the antrum cavity. So this is the complete diagram of the mature Graafian follicle. Now, there are some practice questions also. So section A, multiple choice question. These are the 1 mark question. You have to find out the correct or incorrect statement. So this is the choose the incorrect statement out of this. So this is the practice question. Whenever you get time at home, so read the chapter fully and do all the questions by yourself. The easy questions are mentioned here. So if you get time, you just do these questions also. So these are the wrong statement questions, incorrect statement questions, spot the odd one questions. Now, seminal plasma fluid part is contributed by, so these are some descriptive questions. Which of the following statement is incorrect again? Now, section B, the short answered question, that is of 2 marks question we will talking about. We have just answered the question. The presence and absence of a hammer is not a reliable indicator of vegetative or sexual experience. So, just justify in 2 or 3 lines, not more than that. So we have to justify these questions, we have to answer these questions in just 2 lines or 3 lines. So that is it. Why are the human testes located outside the abdominal cavity? Name the pouch in which they are located. So I have already told you these questions. These particular explanation I have given to you. So corpus luteum is pregnancy. In pregnancy has a long life. Why the corpus luteum is having a long life? So however, if the fertilization does not take place, it remains active only for the 9 days. Why so? So if the fertilization takes place, if the pregnancy occur, it will remain for the longer time. If the pregnancy do not occur, it will remain for the 9 days. So why so? So that it is asking about the justification of these particular short answer type questions. So this is 3 marks question, short answer type question 2, that is write down the significant changes in the secondary follicle. So what are the changes occurring during the formation of the secondary follicle? So you have to write down the complete secondary follicle as well as the diagram. You have to, if you can have the diagram, you can make this, make a label diagram there. What are the events taking place inside the ovary and the uterus during the follicular phase of the menstrual cycle? So the menstrual cycle is divided into the 4 phases, that is the menstrual phase, follicular phase, it is also divided into the ovulation and then after the post-ovulatory phase. So it is asking about the follicular phase, so only one phase it is asking, so need not to mention all the menstrual cycle, you have to mention only one phase, that is the follicular phase. So all the changes, the hormonal changes, the uterine changes as well as the ovarian changes that I have shown you, that I have explained you, you have to mark it there. Now, what is the fetal ejection reflex? Explain in detail. So fetal ejection reflex, so how does the fetal ejection reflex arises, and if you can make the diagram that will be better, but just write down what is the fetal ejection reflex. So how does it arises and how does it affect the ejection of a fetus. Now this is the case based question, on short answer type question, 3 mark question. This is a particular diagrammatic question in which it is a case based question. They will give you a diagram and they can ask you about the particular structure and its details. So the name the stage of the human embryo represented in the given diagram, identify what is A in the figure and mention its function, and mention the fate of inner cell mass after implantation in the uterus. So how does the inner cell mass get transformed into some other structure after the implantation? So you have to write all these. So 1 mark each for, means A, B, C, 3 points are 3 questions are asked from this question, and 1 mark is given for the each particular correct answer. So now the given following diagram is a part of a human female reproductive system. Observe it and give it the answer of the question that follows. So this is the particular structure of the female reproductive system, and you have to answer about X, Y and Z, that is labeled over there, and the 3 question has been raised from this particular structure. Now long answer type question, that is a 5 mark question. Mention the event that lead to the development of placenta during the pregnancy in humans. So you have to write down because of why, because of how the placenta is developed, means what are the structure formed before the placenta and how they convert into the placenta. Explain the role of placenta during the pregnancy, during its action and as an endocrine organ. So we have to write down all the functions of the placenta during the pregnancy, including its role as an endocrine organ. So we have already told you that placenta is a temporary endocrine organ. So what is the function of the placenta as an endocrine organ? So what is gametogenesis? How the maturation phase of the oogenesis is different from the maturation phase of the spermatogenesis? Explain in detail. So all the particular differences I have told you, and I have also told you in detail the spermatogenesis as well as the oogenesis. So you have to write down each and every particular explanation about the maturation phase of the spermatogenesis as well as the oogenesis. Now this is all about